All right, welcome back, everybody. My name is Kevin James. I'm a professor in the Department of Biomedical Engineering here at the University of Virginia, and it is my pleasure to host the second half of this session for knowledge-guided machine learning and translational biology. And I'm happy to introduce our first speaker, Dr. Jisoo Park, computational biologist in oncology at the Genomics Institute of Novartis. So Jisoo is our first industry speaker at this year's workshop. Thank you very much to her employer for allowing her to come and speak. And before Jisoo begins, I'm going to remind that we're doing question and answer through Slido. So please connect on the link. If you have questions, you can ask them of Jisoo in real time, and then we'll address them at the end of her talk. And so Jisoo, floor is yours. You want to begin sharing your screen?
All right, thanks for the introduction. Do you see my slide without a problem?
Looks terrific.
All right, then I'll take it away. Thank you. Hello, everyone. My name is Jisoo Park. And as introduced, I'm a senior computational biologist at Novartis. And I'm going to introduce my postdoctoral study that I published last year with Trey I. Decker at UCSD with the title of Decoding Cancer Cell Maps to Guide Precision Medicine. This work started to tackle the problem that only a small portion of the drugs that go into the clinical trials is eventually approved by FDA. The low success rate of the clinical trials is often due to the incomplete understanding of the mechanisms governing the drug response or improving the cell sensitivity to drugs. To solve this problem, we decided to utilize the deep learning models with the super predictive power, as many researchers have been making a great deal of interest in applying advances in artificial intelligence to tackle various problems in biomedicine. For example, like the deep learning models in radiology. In 2019, deep learning models started outperforming human radiologists in diagnosing various lung diseases based on X-ray images. And more importantly, the models also accurately localized the diseases in the images. The ability of the deep learning model for localizing the disease in the images is very important because it explains why the model made such a prediction, and that information is crucial for doctors to decide the treatment options. And this is exactly what we try to achieve in the context of cancer genomics, to predict the drug response and explain the mechanisms underlying it in terms of the cell mutations. However, the problem gets more complicated when we enter the world of cancer genomics because the patient tumors are very, very heterogeneous, as shown in the scatter plot. It shows a portion of chromosome 17. The scatter plot shows a portion of chromosome 17, which is on x-axis, and then you can see the patients on y-axis has mutations in different genes except TP53. TP53 is shared by so many patients, and the fact makes TP53 a bad pattern to stratify the patient tumors. In addition, there are more complicated problems that the driver mutations for each tumor are known to be different, and cancer pathogenesis is a very complex process where various mechanisms work in a combinatorial way. For these reasons, it's almost infeasible to identify shared patterns among different cancer populations or cancer subtype populations using individual mutations, and therefore performing such an analysis at the pathway level should be the way to go. So to implement the whole idea, we came up with the deep learning-based framework, which enables the analysis of cell mutations at the pathway level. We call this framework DrugCell, and I will introduce it in the following order. I will first present the overview of the DrugCell's framework and show you the predictive power of DrugCell. Then I will present our proof of concept analysis using paclitaxel and how we interpreted the model's prediction. And then I will talk about two studies that validate our findings via DrugCell. First of all, this is the overview of the model structure. Unlike conventional deep learning models, DrugCell model has two branches. One branch on the left takes genotype of the cancer cell lines defined by mutations, and the other on the right takes chemical structure of drugs as an input. Two branches are wired together in a shallow, fully connected neural network to combine the information learned in two separate branches to make a final prediction for drug response. The interpretability of the model's prediction comes from the genotype side of the model, to which we imposed a structural restriction to make it mirror a hierarchical organization of cell functions and becomes biologically meaningful. We used the gene ontology as the hierarchy. Each cellular function or pathway in the hierarchy is assigned with a fixed number of neurons. And instead of fully connecting all neurons between layers, we determine the connectivity between neurons representing each pathway based on the hierarchical relationship between the pathways as defined in the gene ontology. Have this framework implemented, we trained a model using about 500,000 data points for drug response between 1235 cancer cell lines and 684 drugs. The training data is extracted from the combination of the databases curated in Genomics of Drug Sensitivity in Cancer, aka GDSC, and Cancer Therapeutics Response Portal, aka CTRP version 2. The drug response is measured using the area under the dose response curve, which I will refer to as AUC from now on. The example of the training data is shown on the slide. Each cell line is translated into a binary vector representing the mutation status of 3008 most frequently mutated genes across all cell lines. Each SMILES representation of the drug is translated into Morgan fingerprint, a binary vector whose length is 2048. Then the pair of the binary vectors are fed into the model with the drug response AUC values. Then we evaluated the predictive performance of the DrugCell model. The accuracy of our prediction was measured using the Spearman rho between the actual drug response AUC and the predicted AUC, and it was 0.8, as you can see in the first plot. You can also see that the distribution of the model's prediction has a good linear relationship with that of actual drug response. The second waterfall plot shows the list of drugs sorted based on the per drug performance. The red area is the drugs with the Spearman rho bigger than 0.5, which are used for most downstream analysis moving forward. And instead shows the top 10 drugs with the DrugCell's high prediction performance, and those cover drugs in different target classes. When I compared DrugCell's performance to 3 other baseline models, ElasticNet, which is a linear regression model, black box model with a matched number of neurons, and another black box model using a tissue type as additional feature. You can see the DrugCell outperforms the other models for many drugs in the scatter plot. In each plot, each data points represent the individual drug, and the points above the diagonal line are the drugs that the DrugCell could make more accurate prediction than the other model in comparison. After evaluating the accuracy of the DrugCell's prediction as a proof of concept, we analyzed the DrugCell's prediction and interpretation for a microtubule stabilizer, paclitaxel. The predictive performance of DrugCell for paclitaxel was 0.79 in Spearman rho. Then the question now is how the DrugCell model made the predictions for paclitaxel, and in other words, what is the interpretation of the model's prediction? To answer to that question, we used the metric defined in the previous publication of the IDECA lab called relative local improvement of predictive power, which I will call OLIP for the rest of my presentation. Olive score compares the predictive power of the neuron values assigned to a pathway to that of the concatenation of the neuron values assigned to each children. The parent-children relationship is defined in the genontology again. To calculate the Olive scores for one pathway, I first trained a linear model using the neuron values assigned to the pathway. Then I collect the neuron values assigned to all of its children pathways and concatenate them, and then train another linear model using the concatenation. Then the Olive score is defined as the ratio of spearmint row of the parent linear model to that of children linear model. Olive score larger than 1 reflects the state of the parent pathway represented by its neuron values, has more predictive power than for the drug response than the mere concatenation of the state of its children. And it indicates the importance of the parent pathway in the drug cells learning. So going back to paclitaxel, I calculated the Olive scores for all pathways in our genontology and sorted the pathway based on the Olive scores. The noticeable observation that we made is that there were many metabolic pathways within the top 5% of the pathways. So we examined the connection between paclitaxel and the metabolic pathway by treating a 4 to 7 lung cancer cell line with paclitaxel and a glycolysis inhibitor to deoxyglucose and compared the cell viability after treatment. The white box is the cell viability distribution when only the vehicle is injected to the cell line, and blue box is when treated only with the paclitaxel, and gray box is cells treated with the to deoxyglucose, and finally the darker blue is the cells treated with the combination of paclitaxel and to deoxyglucose. The plot shows that the more cancer cells died when treated with the combination of both treatments than when treated with an individual drug, indicating the synergy between paclitaxel and to deoxyglucose. Based on the observation in the paclitaxel analysis, we concluded that the genotype branch of the model learns synergistic targets of the drugs in examination. Then we dived into the question why the model is learning about synergistic targets of the drugs in the genotype branch. To find the answer to that question, we examined the inner working of the DrugCell model more closely. We first looked at the what the drug embeddings that DrugCell learned recapitulate. We performed the PCA on the top level of drug embeddings and visually inspected the data points in two-dimensional space. And then we observed that the drugs that are in the same target classes are well clustered together in that space. This means that the drug embeddings are learning about the mechanism of actions of each drug. Now moved on to the genotype branch, we try to answer to the question, how does a drug cell learn about the synergistic drug target? Considering the synergistic relationship between the drugs and the high Olive score pathways, we concluded that the model's inner working mirrors very well the parallel pathway inhibition theory of synergy. The parallel pathway inhibition theory holds two drugs are synergistic when they inhibit separate pathways that regulate a common essential function. So we hypothesized the biological activity of primary drug is learned by the drug embedding branch, and the parallel pathways are learned by the genotype embedding branch, where the secondary drug's activity is simulated by existing loss of function mutations in each cell. To systematically validate the DrugCell's findings about the synergistic targets made using Olive score, we did the following analysis using a public database called DeepSynergy, which collects synergy scores of drug combinations between 32 drugs. For 25 primary drugs, both existing in DeepSynergy and our training data, we ran the Olive score analysis. And using the top 5 and bottom 5 Olive pathways, we for each primary drug, we identified the potentially synergistic and non-synergistic secondary drugs and compiled the two sets of drug combinations. Our expectation was that the drug combinations compiled using the top 5 OLIP pathways would have higher synergy scores than the one compiled using the bottom 5 OLIP pathways. And as we expected, we observed that the distribution of the synergy scores of the drug combinations compiled using the top 5 OLIP pathways are shifted up compared to bottom 5, and all the rest combinations are existing in the DeepSynergy databases with the statistical significance. There are a few more experimental validation for the DrugCell's finding conducted using the cell lines, but in the interest of time, I will skip them in this presentation. Please find more information about those validation studies in our paper. With that said, I'm going to shift gears towards the very important evaluation for the model trained using the cell lines, which is whether the model can be generalized to other preclinical models or even patient data. To perform such a validation, I assessed the data in PDX encyclopedia, where there are 399 PDX tumors of various tissue types, screened against 40 monotherapies and 27 combination therapies. First, we directly used the DrugCell model, again pre-trained using the cell line data, and predicted the response for the PDX models to the monotherapies. Since the drug response is measured using progression-free survival in the PDX data, which is different from our measurement, which was AUC, we first stratified the PDX models into DrugCell positive group that are sensitive to the treatment and DrugCell negative groups that are resistant to the monotherapy based on the DrugCell's prediction. The binarization is done using the median predicted drug response AUC value. Then I compare the median progression-free survival rate between the two groups. The DrugCell positive group had a longer median progression-free survival rate than the negative group. And when we also compare the distribution of the progression-free survival rate between two groups using log-ranked tests, the two distributions were different with the statistical significance. Then we went ahead and evaluated the DrugCell's ability to suggest effective drug combinations for the PDX models. To do so, we performed the OLIP score analysis for 6 primary drugs that are included in both PDX data for combination therapies in our training data, and evaluated the efficacy of the suggested drug combinations by the DrugCell. To determine the threshold for the percentage of the top pathways suggesting synergistic drug combinations, we performed a ROC curve analysis with the sweeping the portion of pathways to use for the suggestion. As a result, top 20% of the OLIP pathways were used as synergistic targets, and all the rest pathways were considered as non-synergistic targets. The contingency table counts the number of the effective and ineffective drug combinations defined by our model and in the data. The right plot shows an example of effective combinations predicted by DrugCell, BKM120 and encrofenib, BKM20 and encrofenib, and Abraxane and gemcitabine. You can see the progression survival rates were improved for PDX models that received both treatments than the PDX models that received just one of the treatments. On the other hand, for the drug pairs predicted as ineffective by DrugCell, for example, paclitaxel and LCL161 and INC280 and trastuzumab, we found that the progression free survival rate didn't really differ between the PDX model that received both treatments and those that received just one treatment. Finally, we sought to evaluate whether DrugCell can be used clinically to stratify the patient tumors into responsive and non-responsive groups. Using a breast cancer cohort in AACR Genie project containing 221 ER-positive metastatic breast cancer patients who had undergone multiple rounds of therapy, including mTOR inhibitor and CDK4/6 inhibitor. Treatment outcome was measured using overall survival rate in this data set. Using the pre-trained DrugCell model, we predicted sensitivity of breast cancer patient to either mTOR inhibitor or CDK4/6 inhibitor, and stratified the patient into sensitive, DrugCell positive, and resistant, DrugCell negative groups, again, using the median predicted AUC as a threshold. We then compared the distribution of the overall survival rates of the sensitive and resistant patient population that DrugCell predicted. As you can see in the plot, the predicted sensitive patient group has statistically significant longer survivor than the resistant group. When we looked at the specific genetic alterations within the sensitive group and resistant patient population predicted by DrugCell, the sensitive group is much more likely to harbor AKT1 mutations than the resistant group. However, the resistant group had mutations in various genes previously known to be associated with the drug resistant, including ESR1 and then RB1 and P10. And this suggests that the DrugCell stratified the resistant group based on the complex pattern of mutations. And interestingly, for the sensitive group, AKT mutation status was not the best predictive of the therapeutic response. If you look at the plot on the right, AKT mutant patients actually had shorter median survivor rate, and the difference between the overall distribution was not statistically significant. This implies that DrugCell's prediction for the sensitive group is also based on the combination of mutations rather than the status of a single gene. So this analysis illustrates how DrugCell model can be used to effectively guide clinical treatment decisions with significantly greater precision than the single gene marker studies. And I also performed the Olive score analysis for mTOR inhibitor and CDK4/6 inhibitors. The analysis suggested that the two treatments are potentially synergistic pairs, as they prioritize CDK activity for mTOR inhibitor and TOR signaling pathway for CDK4/6 inhibitor. But this Genie data doesn't have a proper drug response information for the combination of two. We examined their synergy using the PDX model data that I previously showed. In the right plot, you can see the progression survival rate of the PDX models improve when treated with the combination of mTOR and CDK4/6 inhibitors compared to when treated only with each treatment. With all that said, let me summarize what I have presented so far. Today, I introduced an interpretable deep learning model for drug response prediction and have shown that its predictive performance was not compromised too much to gain the interpretability. And the interpretation of the model highlights the synergistic drug combinations, and relevant findings are validated using the preclinical models and patient population data as well, indicating the model's generalizability. So I hope I convinced you that the DrugCell has great potential to aid the improvement of clinical trials. And where do we go from here? I will quickly cover a few of the future directions. We can first think about to improve the DrugCell model. We can first think about the incorporation of the other features of the cells and drugs, including, for example, for cell genotypes, we can consider expression profiles or epigenetic states or microenvironmental influences. And definitely secondly, we can think about improving the hierarchy of the cell functions embedded in the DrugCell to make the interpretation more seamless and more biologically meaningful. Lastly, DrugCell can only learn for the drugs with the known chemical structures, but the framework can be adapted and modified to learn about the therapies without known chemical structure as well. But that's going to be more of a longer term open question. Lastly, I will acknowledge everyone in the IDEC lab, especially Trey Idecker, my postdoc supervisor, and Brent Quenzi, who is the first author of the DrugCell paper. And I thank our funding agencies. And with that, I will take questions.
Excellent. Thank you very much, Jisoo, for an outstanding talk on knowledge-guided machine learning applied to translational biology from cell lines to patients. There was one question that came up in Slido. It was relatively early, and it says the predicted drug response in Spearman is Spearman correlated. I think this was the original development of DrugCell, but the model appears to overestimate the effect of drugs with an AUC. I guess I measured AUC around 0. Any explanation for this bias?
You mean here?
I perceive so, yes. I
see. Yeah, it's one of the things that we have to also think moving forward to further improve the model's predictive performance, which is that the data, one thing that I should mention, I should tell you about the data itself is that in the data, the training data is pretty imbalanced, that there are more resistant drug response data than the sensitive drug response data. We have discussed a lot about how to resolve the imbalance in the between the sensitive and then the resistant drug response data, but yes, there are, so for the it's well thought and considered that for the sensitive cell response to the drugs, it's a harder problem to predict. That's why it's actually yeah, I'm not sure that it answered the question completely.
Okay, well, no, I think that's good. All right, thank you.